Okay, let's unpack this. We've got a stack of truly fascinating information in front of us, taking a deep dive into something fundamental. What do microbes actually need to eat to survive and grow?
That's right. We're pulling insights directly from a detailed text on microbial nutrition. It covers everything, really, from the absolute basic chemical building blocks,
The real essentials.
exactly, all the way to the surprisingly complex art of trying to feed them in a lab setting and even how they, extract energy from their environment.
Our mission today is to cut through the detail and pull out the most important and hopefully most surprising facts about microbial nutrition. We want to give you a clear understanding of their fundamental requirements,
Yeah.
whether you're thinking about culturing them for something specific or maybe just want to better understand the invisible world that lives everywhere around us and, well, even inside you.
Because you might think feeding microbes is simple, maybe just water and a bit of sugar.
Right. Seems easy.
But as we saw in the material, it quickly gets into some pretty intricate chemistry, requires incredibly tiny amounts of unexpected elements, and involves overcoming some real challenges in the lab.
Challenges like what?
Well, things like essential nutrients reacting
Yeah.
and just dropping out of solution, or even your microbial food turning brown during preparation. Stuff you wouldn't necessarily think about.
Okay. So, where do we start this nutritional journey then?
Well, just like us, microbes need certain elements in pretty substantial quantities.
Big stuff first.
Right. The source calls these macro elements. They're required at concentrations generally higher than 10 to the power of -4 molar, which sounds tiny, but it's actually quite significant in this context.
Okay, 10 to 4 molar. Got it. So, what are the main players?
Well, the absolute top of the list for many microbes, specifically the heterotrophs, the ones that eat organic stuff, is a source of both carbon and energy.
So often the same thing.
Often, yeah. One single compound provides both. The source gives us a simplified way to think about microbial growth. You take your carbon and energy source, add a nitrogen source and other necessary nutrients.
And you get.
And the result is new cell mass plus some byproducts and, heat, like any living process.
It's almost like a recipe. The material even gives us an average approximate formula for bacterial cell mass. CH 0.18, 0.5, N 0.2.
Yeah, it's a kind of average blueprint.
Now, while it does show the general elemental percentages and carbon can be a massive chunk, sometimes over 60% of dry weight, which is huge.
It really is.
The source is really clear. You can't just look at those percentages and mix elements directly. It's not that simple.
Exactly. The form in which these elements are supplied matters immensely, as does the presence of other compounds. It's all about the chemistry.
Okay, so after carbon, what's next?
Following carbon, nitrogen is another absolute cornerstone. It's crucial for building proteins and nucleic acids, DNA, RNA.
The building blocks of life, basically.
Right. Makes up a significant portion, maybe 12 to 15%
Yeah.
of the dry weight of bacteria. And microbes can be, well, a bit picky about how they get it.
Picky how? Different forms.
Different chemical forms, yeah. The ammonium ion, NH4 plus, seems to be the favorite for many.
Why is that?
It's the reduced form, so it's the most energetically efficient to use. They don't have to spend much energy processing it. And nearly all prokaryotes, bacteria, and archaea can just grab it and incorporate it directly.
Okay, so ammonium is easy street. What else?
Organic forms are also good. Things like amino acids, small proteins, or urea, they're readily available sources for most microbes.
Makes sense, already partly processed.
Exactly. Less preferred are the oxidized forms, like nitrate NO3 or nitrite NO2,
Why less preferred?
because these require the microbe to spend energy reducing them first, pulling oxygen off. And actually, many microbes, especially yeast, simply lack the machinery to use nitrate at all.
Okay.
And here's a key point the source makes. Nitrite, NO2, that intermediate form, can actually be toxic to many microbes.
Wow, really? So it's not just less efficient, it can be harmful.
It can be, yeah. And then there's the ultimate source, grabbing nitrogen directly from the air, N2 gas.
Like legumes do with bacteria?
Kinda, yeah. Only a select few prokaryotes, the nitrogen fixers, can do this amazing feat, and it requires a massive energy investment from the cell. It's a tough way to make a living.
Okay, so carbon, nitrogen. What's the next macro element?
Sulfur is next on the list. It's vital for certain amino acids like cysteine and methionine and some coenzymes. Makes up maybe 0.2 to 1% of dry cell mass.
And like nitrogen, does it come in different forms too?
It does. Similar story, really. You've got reduced forms like elemental sulfur or hydrogen sulfide, H2S, organic forms in amino acids, and the oxidized sulfate ion, SO4.
So what do people usually put in lab media?
Sulfate is common, it's stable and easy, but some microbes specifically need those more reduced forms to grow. Again, depends on the bug.
Okay, and here's where the idea of macroelements gets a bit nuanced, which is something I found really interesting in the material. Sodium, Na plus, and chlorine, Cl, salt, basically.
Ah, yes, salt.
The source says they're important for membrane transport, helping things get in and out of the cell, standard stuff. But surprisingly, it says they aren't essential, not in large amounts anyway, for many common lab microbes, like E. coli is the classic example.
Right. For your standard E. coli growing in a rich medium, it doesn't need loads of extra salt added. It gets enough,
That's a big
incidentally.
step, right?
There is. They aren't needed in those macro amounts for most. However, for microbes that live in really salty environments, the halophiles, marine microorganisms.
Things living in the sea or salt lake.
Exactly. Or even some bacteria that live in the gut of cows, the rumen bacteria mentioned in the source. For them, sodium and chlorine are absolutely required macroelements. Their whole biology is built around managing high salt concentrations.
So it really, really depends on the specific micro and its natural home.
Totally. Context is everything in microbiology.
Okay, fascinating. So shifting gears now, from the elements needed in bulk to those required in unbelievably tiny amounts, the microelements or trace elements.
Yeah, the trace stuff. We're talking micrograms or even nanograms per liter. It's almost nothing, but absolutely essential.
The source mentions calcium and iron are sometimes in a gray area, kind of borderline.
Yeah, they're sometimes needed in slightly higher amounts than, say, cobalt or zinc, but still generally much less than the macros. So they sit a bit in the middle sometimes, depending on how you classify them.
And what's their main job, these tiny amounts?
Their main job, as the source explains, is usually acting as inorganic cofactors for enzymes.
Cofactors, like helpers.
Exactly. Think of them like tiny tools or keys that help the enzyme protein do its job, maybe helping it fold correctly or actually perform its specific chemical reaction. Little sparks for the biochemical machinery.
Okay. And the material lists examples.
It does. It mentions a core group, usually required, group A they call it, like calcium, iron, manganese, cobalt, copper, and zinc. Pretty much standard kit for many microbes.
And then there are others.
Yeah, groups B and C mentioned are needed by more specific microbes for particular enzymes. Things like molybdenum, nickel, selenium, tungsten. These often pop up in enzymes involved in, say, metabolism without oxygen in anaerobes.
Right. And iron, Fe, gets a special call out in the source material. Why is that?
Iron. It's a big story. And this is genuinely fascinating, I think. Iron is super abundant on Earth, right? Dirt, rocks.
Yeah, everywhere.
But under aerobic conditions, with air around the more usable form, Fe++ Fe quickly oxidizes to Fe+++++, ferrous to ferric.
And that's the problem.
It's a huge problem because Fe++ is incredibly insoluble at neutral pH. It basically forms rust or other related minerals. It just drops out of solution, making it really hard for microbes to access.
So it's there but locked away. How do they grab hold of this essential metal then?
This is where their biochemical ingenuity really shines. Microbes have evolved to produce these amazing molecules called siderophores.
Siderophores, okay.
The source names different types, hydroxamic acids, phenolic ones, peptide-based ones. What's cool is how they work. They chelate the iron.
Chelate.
Yeah, it means they literally grab onto the Fe+++ ion like a molecular claw, binding it super tightly through multiple chemical bonds.
A molecular claw, that's a great way to picture it.
It is. This keeps the iron soluble outside the cell, and then the microbe has specific transport systems on its surface that recognize the siderophore iron complex and actively pull it inside. It's a very clever scavenging system.
Wow. Okay, that's impressive. And the source mentions another twist here with iron. Some microbes don't need it at all.
Right. Some, like many lactic acid bacteria, the ones that ferment milk into yogurt and cheese,
Yeah.
they often lack the machinery that uses a lot of iron, like the electron transport chain for respiration. And for them, manganese, Mn plus Ang often steps in and substitutes for iron in some of their other essential enzymes.
So manganese does the job instead.
In some cases, yeah. It's like a backup plan nature put in place, or maybe an ancestral state.
Yeah.
It just shows how adaptable microbial biochemistry can be. M plus filling in for Fe.
Very cool. Okay, now, distinct from the elements, the inorganic stuff, the source talks about growth factors. How are they different?
Right. Growth factors are defined differently. They are essential organic compounds, things like vitamins or amino acids.
Stuff made of carbon.
Exactly.
Oh.
Organic molecules that the cell itself cannot synthesize, but it absolutely needs them, usually in tiny quantities. They get incorporated pretty much as is into cell components like building blocks or parts of enzymes.
Mhm.
They aren't typically just broken down for energy like, say, glucose might be.
Ah, okay. So these are like the essential vitamins or specific amino acids we need in our diet because our bodies can't make them from scratch?
That's a perfect analogy. It's exactly the same principle.
And the source groups them.
Yeah, it groups them into four main categories. Amino acids, the building blocks for proteins, purines and pyrimidines, essential for making DNA and RNA.
The genetic material.
Right. Then vitamins, which are often crucial parts of coenzymes, helping enzymes work. And finally, fatty acids and sterols, needed by some microbes without cell walls, like mycoplasmas, or some yeasts and protozoa for their membranes.
And are some microbes needier than others for these?
Oh, definitely. The source notes that some microbes, particularly certain lactic acid bacteria again, are incredibly demanding. They're fastidious is the technical term. They require many different growth factors to be supplied in their medium.
Why is that?
They've essentially lost the ability to synthesize a wide range of these essential compounds themselves over evolutionary time. They rely on finding them ready-made in their environment, like nutrient-rich milk or decaying plant matter.
Okay, so we've got the shopping list, the macroelements, microelements, and these organic growth factors. Now, how do we actually turn this list into a meal? The source dives into formulating culture media.
Making the microbial soup.
Exactly. Which it describes as definitely a blend of art and science.
It really is. It breaks down media types based on what's in them. You have complex media, which use ingredients like yeast extract or beef extract, maybe peptones.
Stuff where you don't know every single molecule.
Precisely.
Mm.
They're great for growing many different things because they're packed with nutrients, but the exact chemical makeup is undefined, unknown. Then you have chemically defined or synthetic media.
And that's the opposite.
Exactly. You know exactly every single chemical component and its precise concentration. You weigh everything out carefully. Much more controlled, but sometimes harder to get growth if you miss something.
And then there are minimal media.
Right. Minimal media contain only the absolute bare necessities that specific microbe cannot make itself. Everything else that has to synthesize from those basic ingredients. Great for genetic or metabolic studies.
And basal media.
Basal media are usually defined media that are deliberately missing one or more specific components.
Yeah.
You use them, for instance, to test if a microbe needs that missing component or can use a certain nutrient.
Okay, different tools for different jobs. Now, preparing these media, especially making sure they're sterile so only your target microbe grows, that sounds like it could have problems.
Oh, it definitely presents several chemical hurdles. The source highlights a few key challenges, especially during heat sterilization, like autoclaving, which uses high-pressure steam.
Okay, like what?
Well, one big one is dealing with heat-sensitive compounds, or heat labile compounds. Some amino acids, it mentions tryptophan, glutamine, asparagine, and many vitamins, like thiamine, riboflavin, pyridoxine. They're simply destroyed by the high heat of autoclaving.
So they're gone before the microbes even get a chance.
Pretty much. The solution here is filter sterilization. You dissolve these delicate compounds separately, pass the solution through a very fine filter, like 0.2 microns, that traps bacteria.
But lets the molecules through.
Exactly. Then you add the sterile solution to the base medium after it's been autoclaved and cooled down.
Clever. What other pitfalls are there?
Another classic lab problem is the Maillard reaction. You might actually know it from cooking.
Browning. Like on toast or grilled meat.
That's the one. Sugars reacting with amino acids or proteins under heat, causing browning. In media preparation, this doesn't just look bad. It actually degrades the sugar, making it less available, and can even produce some inhibitory compounds.
So how do you avoid your microbe food getting toasted?
The fix is usually simple. Autoclave the sugar solution separately, just in water, or even better, filter sterilize the sugar solution too, and add it cold to the autoclaved base.
Okay, keep the sugars and amino acids apart during heating. Got it.
Yep. Ammonium salts also need a bit of care. If the pH is high, say above 7 or 8, during autoclaving, they can lose ammonia gas, NH3. This changes the nitrogen concentration and the pH. So keep the pH lower during autoclaving if you can.
And urea.
Urea is another one that's heat labile. Breaks down easily with heat. So filter sterilize it if it's your primary nitrogen source.
Okay. And here's a really significant headache for media makers that the source emphasizes. Precipitation. Things falling out of solution.
Ah, yes, the bane of many a media prep session.
Remember those essential macro and micro elements? Especially the positively charged ones, the cations?
Like calcium, iron, magnesium.
Yeah, well, these divalent and trivalent ions, K plus A, Fe plus A plus A, Fe plus soy Mg plus plus, and even sometimes monovalent ones like potassium K plus and sodium Na plus, can react with phosphates or hydroxides in the medium,
Especially when heated.
especially when heated at neutral or alkaline pH, like during autoclaving.
Right.
They form insoluble compounds, literally tiny mineral particles, precipitates that settle out.
Like little rocks forming in your nice clear broth.
Exactly. And the problem is, this makes those essential elements unavailable to the microbes, even though you carefully weighed them out and put them in.
So the nutrients are there, but the microbes can't get them.
Right. The source gives examples like magnesium sodium phosphate or magnesium potassium phosphate, magnesium ammonium phosphate. These can all precipitate out. And phosphates can apparently also complex with glucose if autoclaved together at higher pH.
Wow. Okay. Lots of unwanted reactions. So what are the solutions?
A few tricks. You can try lowering the pH during autoclaving, maybe around 6 or 6.5, as things tend to be more soluble in slightly acidic conditions. Or you can autoclave tricky components separately.
Like the magnesium and the phosphate solution.
Exactly. Autoclave them in separate flasks and then combine them aseptically when they're cool. That often prevents the precipitation.
Makes sense. And trace metals face the same issue.
Yep. Trace metals like iron face the same precipitation problem, especially at neutral pH where Fe+++ is so insoluble.
And the clever solution here, which we touched on with iron uptake, is using chelating agents, right?
Exactly. The source brings them up again in the context of media prep. These are molecules that bind tightly to the metal ions, like our molecular claw for iron.
Holding them in solution.
Precisely. Keeping them soluble and available in the solution, preventing them from reacting with phosphate or hydroxide and precipitating out.
Do complex media ingredients help with this?
Yeah, the source mentions that complex media often have things like amino acids or peptides that naturally have some chelating action, which helps keep metals soluble.
But in chemically defined media, you might need to add something specific.
Right. Where everything is pure chemicals, this problem can be more acute, so you might need to add a specific chelator. The source mentions EDTA, which is ethylenediamine etracetic acid.
A common lab chemical.
Very common, very effective at binding metals. But the source adds a caution. Sometimes EDTA can bind the metal too tightly, making it unavailable to the microbe even though it's soluble. Over-chelation, basically.
Ah, okay, so it holds on too tight, yeah.
Yeah.
Citric acid is often mentioned as a good alternative. It chelates metals like iron well enough to keep them soluble at neutral pH, but maybe not so strongly that the microbe can't get the metal off it later. It provides just the right level of binding, often.
Finding that chemical sweet spot. Okay, so we've covered what microbes need, the elements, the factors, and the chemistry of actually making their food without it falling apart or browning.
Yeah, the practicalities.
But how do they actually get the energy and carbon they need to build themselves and grow? The source explains that this fundamental choice or capability defines their nutritional class or lifestyle.
Right, it's like classifying them based on their energy and building material sources.
It breaks it down into categories based on, well, three main things.
Yeah.
Where they get energy, where they get electrons.
Or hydrogen atoms, essentially, which carry the electrons.
Right, and where they get their carbon.
Okay, so let's break that down. For the energy source.
Two main groups. Phototrophs use light energy photosynthesis. And chemotrophs get energy from chemical reactions, specifically oxidation-reduction reactions, redox reactions.
Okay, light eaters and chemical eaters.
Huh.
What about the electron source?
Again, two groups. Organotrophs use organic compounds as their electron donor. Think sugars, fats, proteins. Lithotrophs use inorganic compounds,
Inorganic. Like what?
like hydrogen gas, H2, hydrogen sulfide, H2S, ammonia, NH3, or even ferrous iron, Fe2+. They can literally pull electrons off rocks or simple inorganic molecules.
Wow, okay. Eating rocks, almost. And the third category, carbon source.
This one might be more familiar. Heterotrophs get their carbon from preformed organic compounds like sugars, amino acids. They eat things made by others. Autotrophs are the self-feeders.
They make their own food.
Essentially, yes. They fix carbon dioxide, CO2, from the atmosphere or dissolved in water, building their own organic carbon molecules from scratch, using energy from light or chemical reactions, like plants do.
Right. So the source combines these terms.
It does. It combines them to give us the standard terminology you might hear in microbiology. You get things like photolithotroph.
Okay, photo means light. Litho means inorganic electron donor. So They'd probably fix CO2 too. An autotroph.
Usually, yeah. So light energy inorganic electrons fixes CO2.
Hmm.
Think cyanobacteria, purple sulfur bacteria,
Hmm.
then photoorganotroph.
Light energy, organic electrons. Probably uses organic carbon too. Heterotroph.
Often, yes. Light energy, organic electrons, organic carbon source. Some photosynthetic bacteria fall here. Then chemolithotroph.
Chemical energy, inorganic electrons.
Yeah.
Likely fixes CO2.
Correct. Chemical energy, inorganic electrons, fixes CO2. Think bacteria that oxidize ammonia, sulfur, or iron. Really important in nutrient cycling.
Yeah.
And finally, chemoorganotroph.
Chemical energy, organic electrons,
Ah.
and uses organic carbon. Heterotroph.
That's the one. Chemical energy, organic electrons, organic carbon. That's us. And most of the bacteria we commonly think about, especially pathogens and fungi.
Okay, that four-part system makes sense.
Yeah, the source prefers that system, although it mentions that sometimes people simplify it by linking lithotrophy directly to autotrophy and organotrophy to heterotrophy. That gives you terms like photoautotroph or chemoheterotroph.
Which is maybe less precise but quicker to say.
Maybe. The source includes a really helpful summary table to map all these combinations out, which is great.
Good. And did it mention the types of energy metabolism?
It did, briefly. It touched on the core ways chemotrophs get energy. Respiration, which uses an external, usually inorganic electron acceptor.
Like oxygen.
Like oxygen in aerobic respiration, yes. Or in anaerobic respiration, things like nitrate or sulfate or even carbonate can be the final electron acceptor instead of oxygen.
For microbes living without air.
Exactly. Then there's fermentation. This is different. Here they use an organic compound as both the electron donor and the electron acceptor. They're basically just shuffling electrons and protons around internally on organic molecules, releasing a bit of energy, no external acceptor needed.
Like making yogurt or beer.
Yep, classic fermentations. And of course, photosynthesis for the phototrophs, converting light energy into chemical energy like ATP.
Okay, a whole range of energy strategies. Now, what about the environment itself? Did the source talk about controlling conditions?
It did. It brings up something absolutely critical that can make or break microbial growth, even if all the nutrients are perfectly provided. pH control.
Keeping the acidity or alkalinity stable.
Exactly. Microbial activity itself often changes the pH of their environment, their growth medium, and keeping that pH stable within the range the microbe likes is vital.
Why does the pH change? Just from them growing?
Yeah, from their metabolism. The source lists a few key reasons. pH can become more acidic if the microbe takes up ammonium ions, NH4+, because when they take the NH4+ in, they often release a proton, H+, out to maintain charge balance.
Okay, uptake of a base releases acid.
Right. Or, more obviously, if they produce organic acids as metabolic byproducts, like lactic acid bacteria making lactic acid. That directly lowers the pH.
Makes sense. And how can pH go up? Become more alkaline?
That can happen if they take up amino acids and then cleave off the amino group, deamination, which can release ammonia or related basic compounds, or if they consume acid anions.
Like eating acetate or nitrate.
Exactly. Consuming an acid makes the environment less acidic, so the pH goes up. Also, if the microbe has the enzyme urease and urea is present in the medium, breaking down urea releases ammonia, which is basic, and drives the pH up quite strongly.
Okay, so their own eating habits change their environment's pH. How do you control that?
Well, in big industrial bioreactors or fermenters, you might use sophisticated automated systems. pH probes constantly measure the pH, and pumps automatically add acid or base as needed to keep it exactly where you want it.
That in a regular lab flask, or a test tube, or an agar plate.
Yeah, in standard lab cultures like broth or agar plates, you can't really do that titration easily. So you must include a buffer in the medium itself.
Something to resist those pH changes.
Precisely. And the source really focuses on phosphate buffers, which are super common in microbiology, usually made from a mix of potassium dihydrogen phosphate, KH2PO4, and dipotassium hydrogen phosphate, K2HPO4.
Why are they so popular?
Well, they have advantages. They buffer effectively in the pH range most microbes prefer, roughly pH 6 to 8. They're generally non-toxic. And bonus, they provide essential phosphorus P and potassium K as nutrients.
Sounds ideal. Any downsides?
The big disadvantage we already mentioned, they can precipitate with those tricky cations like magnesium and calcium, especially during autoclaving at neutral pH.
Yes, the precipitation problem again.
The material references the chemistry here. Phosphoric acid is triprotic, meaning it can lose 3 protons, H+. The relevant step for buffering around neutral pH involves the H2PO4 ion losing a proton to become HPO4. This equilibrium has a pKa value around 7.2.
And the pKa is important because
The pKa is like the pH sweet spot for a buffer. It's the pH where the buffer is exactly 50% in its acid form, H2PO4, and 50% in its salt-based form, HPO4. That's where it's most effective at resisting changes in pH when you add small amounts of acid or base.
so it works best around pH 7.2.
It works best at pH 7.2, and it's generally effective within about plus or minus 1 pH unit from the pKa, so roughly pH 6.2 to 8.2 for phosphate, which covers a lot of microbes.
Okay. Did it mention other buffers?
It did, briefly. Things like adding calcium carbonate, calicrete powder to agar media for microbes that produce a lot of acid. The acid dissolves the chalk, neutralizing it. Also, chemical buffers like phthalic acid, used as potassium hydrogen phthalate, KHP, which buffers down around pH 5.5. Or specialized buffers like MES buffer for the pH 5-7 range.
And cautions.
Yeah, it cautions that many organic acid buffers, while they might have useful pKa values, can actually be toxic or inhibitory to microbes, especially at lower pH values where the undissociated acid form can cross cell membranes more easily.
So phosphate is often the safer bet, despite the precipitation issue.
Often, yes. And the source even shows the underlying chemistry for actually calculating how to make a buffer of a specific pH and concentration using the Henderson-Hasselbalch equation.
The classic buffer equation.
Yes, pH equals the pKa plus the logarithm of the ratio of the concentration of the salt form, the conjugate base, divided by the concentration of the acid form.
So lets you figure out the proportions needed.
Exactly. It walks you through an example. Preparing 1 liter of a 0.45 molar potassium phosphate buffer at pH 7.5. You plug pH 7.5 and the pK of 7.21 into the equation.
And solve for the ratio of HPO4 to H2PO4.
Right, and you find that for pH 7.5, you need that ratio to be about 1.95. So roughly twice as much of the salt form, T2HPO4, as the acid form, KH2PO4.
Okay, you've got the ratio. How do you get the actual amounts?
Well, you know the total phosphate concentration needs to be 0.45 molar, and you know the ratio of the two forms needs to be 1.95 to 1. So you just divide that total concentration according to that ratio. You calculate that you need about 0.296 molar of the K2HPO4 and about 0.154 molar of the KH2PO4.
And then you just weigh out the grams needed for those molar amounts in 1 liter.
Exactly. While the source admits that in practice, many labs might just weigh out roughly the salts and then adjust the final pH with concentrated acid or base using a pH meter.
Which is probably quicker day to day.
Probably. But understanding the Henderson-Hasselbalch calculation reveals the precise chemistry required to actually achieve that stable pH environment you're aiming for. It shows what's really going on chemically.
It's a great example, actually, of how deep the chemistry goes, just to provide a comfortable, stable living space for these tiny, tiny organisms.
Wow, that truly was a deep dive into the intricate world of microbial nutrition. We've really covered a lot. We've gone from the basic pantry list of elements, the macros and micros,
The essential organic extras, the growth factors.
through the significant chemical challenges of just preparing their food in a lab without it, precipitating or degrading.
The practical chemistry hurdle.
Explored the really diverse ways they power themselves, getting energy and carbon.
Phototrophs, chemotrophs, lithotrophs,
Yeah.
the whole spectrum.
And finally, wrestled with the critical importance of controlling their immediate environment, especially pH, through buffering.
Yeah, understanding these fundamental requirements isn't just academic, is it? It's absolutely crucial for countless real-world applications.
Like what?
Well, everything from diagnosing diseases by being able to grow the specific pathogen, to environmental cleanup using microbes that eat pollutants, to huge industrial biotechnology fermentations producing everything from enzymes and vitamins to biofuels and medicines.
It all comes back to feeding them right.
It really does, and it underscores the incredible complexity and, frankly, the biochemical sophistication packed into these microscopic lives. They're not simple bags of chemicals.
So, thinking about all of this detailed knowledge about their needs, the incredibly specific cocktail of macro elements, those vital trace metals, delicate growth factors, and that carefully managed pH needed for different microbes to thrive. How does this change your perspective maybe on the unseen world living all around us, in the dirt, the water, even, in our own bodies?
It makes you realize it's not just a random soup out there. It's highly structured niches.
Mm-hmm.
And considering the precision outlined here, the exact elements needed, sometimes in vanishingly small trace amounts, the need for things like natural chelating agents perhaps, the specific buffering requirements.
Yeah.
It poses a question, doesn't it?
Which is,
What unexpected natural environments out there might actually be providing the perfect, naturally formulated pH-buffered buffet for specific, maybe currently unknown, types of microbes, allowing them to flourish in ways we can barely replicate, or perhaps haven't even discovered yet, in a lab?